learning objectives include nucleic acid-based microbial identification, and we'll study PCR. PCR basically is polymerase chain reaction. Basis of PCR is very simple. All is need is a pair of primers. So as we saw that antibodies provide specificity in serological testing, serological interactions. Nucleic acid-based testing requires a piece of DNA, which we call a signature DNA or signature sequence, because that sequence has to be specific to the organisms. It should not be shared by any other organisms for microbial identification. Similarly, PCR, we designed primers that basically target a fragment of DNA which is unique to the organisms. So it is also called a signature sequence. So we target in PCR a signature sequence not shared by any other organism. And the technique has 3 steps basically. We start with the double-stranded DNA, like in this case here. This is our target DNA or signature sequence which we want to amplify. What we would do is we would separate these. It's a double-stranded DNA. We would separate double-stranded DNA into single-stranded DNA by heating, in this case, PCR. And this stage, the first step, is called denaturation stage. And we would heat the reactants at 94 or 95 degrees Celsius for a minute or so. Then we add primers into this PCR tube. The primer basically are the ones that provide the specificity for the detection. So the primers are designed in a way that they target the signature sequence. When we lower the temperature after denaturation step, the primers, because they're a lot more in concentration, so they would attach to these target DNAs, and the process or the step is called annealing stage or annealing process. Typically, the temperature is 50 to 56 degrees Celsius. In the next step, we would allow these primers to be extended. And the reason it is DNA is made, new DNA molecules are made, it's called extension or extending stage. And the DNA polymerase that we use, it typically requires 72 degrees Celsius temperature. So double-stranded DNA is denatured, made single-stranded DNA, primers are annealed, and then the primers are extended to make new copies of DNA. 30 or so cycles are achieved or carried out by PCR machine automatically. At the end of the 30 cycles or 35 cycles, enough target DNA accumulates that we can detect on the gel. So after the PCR cycles are over, we put our DNA, amplified DNA, through the gel, and then we detect specific bands. The presence of these bands, as you could see here, it shows that the specific DNA is there. That means specific microorganism is there. It's a wonderful technique, very rapid and very precise, and could be performed in less than 3 hours. There is another modification these days available, what we call real-time PCR, which is even a lot quicker. But the basic principle is the same. So in summary, PCR provides a quicker way of microbial identification.